Very good afternoon, everybody. Good afternoon. Right. Uh, right. Okay. Today, today we discuss the library, genomic library, and cDNA library, genomics and cDNA library. So, what is here? Types of DNA library and cDNA library. What is the difference between the genomic library and cDNA library? We are to elaborate here one by one. We are to discuss here. So, what is genomic means DNA library? Here, the genomic information can be obtained by two primary methods are there. Based on this, DNA libraries are divided into two types. That is the genomic and cDNA library. So here, what is the genomic library? Here, genomic library. A genomic DNA library is collected means a collection of DNA fragments that represent all genetic information of an organism. This includes both coding and non-coding regions of DNA. There is means here the DNA fragments, small fragments, so which is represent the genetic information of that particular organism. So, which are the coding and non-coding regions are there, that fragment. So, this one is called the genomic libraries. The libraries are suitable for a wide range of applications, including genome mapping and comparative genomics. It allows the study of regulatory elements and non-coding sequences that are important in gene expression and regulation. Okay, so this one is the most important. Genomic library is the most important in the that is the study about the regulatory level. The genomic libraries also have certain disadvantages, include the complexity and size of handling large DNA fragments and the resource intensive process of patterning and maintaining the such library. So here, wait, I show the figure here. So see here. So see this picture. That is the cDNA library means the complementary library. That is nothing but cDNA means the complementary. So it is a collection of cDNA molecule derived from the mRNA. So means unlike genomic libraries, cDNA libraries represent only the expressed genes of an organism, excluding non-expressed genomic regions such as introns and other non-coding sequences. The cDNA libraries are useful for studying the gene expression, protein function, producing a recombinant protein. Since cDNA libraries exclude non-coding regions, they provide a more focused view of the expressed genetic information. So this one is more important. And another one is a disadvantage of cDNA library include the limitation of studying gene regulation due to the absence of regulatory sequences, limited gene diversity, and bias towards the highly expressed genes. The cDNA libraries are specifically created from eukaryotes to study the expressed genes. The prokaryotes do not contain introns, therefore, creating cDNA libraries for prokaryotes is generally not necessary as their genomic DNA directly corresponds to their mRNA. So, what is the difference between the cDNA and genomic library? So, we have to discuss the some difference. What are the differences here? So, first one is the content. What is the content here? The genomic libraries contain DNA fragments that represent the entire genome of an organism. So means here that each fragment is represent total genome of an particular organism. So cDNA means complementary DNA libraries consist of only the cDNA sequence that are actually expressed. Means complementary DNA. Okay, that particular cDNA is actively expressed. The size of DNA fragment. Another character is the size of the DNA fragments. The DNA fragments are larger, ranging from thousand to million base pairs are there. Very large in size, genomic library. But the cDNA libraries contain smaller DNA fragments, very small in the complementary DNA. So coding and non-coding regions. Next character is the coding and non-coding regions. It contains both coding and non-coding region, including introns and regulatory sequences present in the genomic library. In the cDNA libraries, it contains only coding regions or expressed genes only. Another character, source of DNA. The source of DNA is the total genomic DNA extracted from cell or tissue. So here the total genome is from that DNA which is obtained from a particular cell or tissue. Here in the cDNA libraries, there is a source of DNA from a cDNA library is mRNA which is converted to cDNA. From that mRNA then converted into cDNA. Okay. And the vectors used. What type of vectors here? It is uses vectors such as bacteriophages, bacterial artificial chromosomes, which can hold large DNA fragments. Here in the cDNA libraries, what vectors we are using here? Plasmids, which are suitable for the smaller cDNA fragments. So in the plasmids, we are to use here cDNA libraries, but the bacteriophages are used in the genomic library. And another one is application. Another character is the application. So genomic libraries are used for the genomic mapping, comparative genomics, and studying gene regulations. So total genomic libraries, we have to study the all the characters means gene mapping, comparative, and what type of the gene regulations we have to study in the genomic library. But the DNA libraries that particularly used for studying the gene expression pattern and function of gene that express gene expression regulations and what is the function of expressive genes only. Here we are to study the cDNA, means the comprehensive DNA. Okay, that is the difference between the genomic libraries and cDNA libraries. Okay, so another that is character, so that is the construction means of genomic library. The first one is the isolation of genomic DNA, fragmentation of genomic DNA, and cloning, transformation. That is. So first one is the construction of that is of genomic library, isolation of genomic DNA. The construction of genomic library begins with the isolation of genomic DNA from the organisms of interest. So here the genomic library, which is the isolated of cDNA, that particular organism. Genomic DNA can be isolated using different methods such as cell lysis, protein digestion, and phenol chloroform extraction. And the isolated DNA represent the entire genome of the organism and contain both coding and non-coding regions. So here, that is the first step of the isolation of the genomic DNA. And coming to the fragmentation of the genomic DNA. So isolated genomic DNA obtained is often too large to be cloned directly into vectors. So it is need to be fragmented into smaller fragments suitable for the cloning technique. So after isolation, what happened here? After isolation of the genome DNA, then we are to fragment it, that genomic library. After isolated, so large that when we clone directly into the vector. Okay. So after that one, this is the transfer fragmentation can be achieved using the physical methods such as uh, what is that one? Sonication, mechanical shearing, or enzymatic method involving the restriction enzyme. So third one is the cloning. The fragmented genomic DNA is then cloned into a suitable vector. The fragmented genome is then cloned into the suitable vector. Some common vectors are used for the genomic library. There is the construction include plasmids, bacteriophages, bacterial artificial chromosomes, and yeast artificial chromosomes. These are used for the vector to use into the cloning. The bacterial vectors are suitable for smaller DNA fragments, while yeast artificial chromosomes are used for the larger DNA fragments. Okay. The vectors are treated with the restriction enzymes to create sticky ends that are compatible with the fragmented DNA. DNA ligases, that ligase enzyme is used to bind the DNA fragments to the vector, creating a recombination DNA molecule. నాకు అరే డ్రాయింగ్ కాదురా, ఇది వైఫై నాది కనెక్ట్ అవుతాందిరా. నాకు తెలియదురా ఇక్కడ వైఫై ఎందుకు ఆన్ చేసిన దీన్ని. ఇగో నువ్వు పండుకోపో ఫస్ట్. నాకు క్లాస్ ఉన్నదిరా, నాకు వదిలేయకురా, కూడా ఇక మళ్ళా నీళ్ళని కూడా కష్టపడ్డది ఫ్యామిలీ చెప్తున్నా. ఆర్డర్ అయ్యి చక్క పండుకోపోతుంది. నాకు అయిపోద్దిరా. So next one is the transformation. So the transformation means the recombinant vector containing the cloned genomic DNA fragment are transferred into the suitable host, or which is already we are to selected that to mainly usually E. coli or yeast we are to use as a host organism. So the transferred host cells take up the recombinant vectors and are cultured on agar plates containing selective media to allow the growth of the colonies containing the recombinant DNA. These colonies form a genomic DNA library. So here, okay, I show to another diagram that way. This one is the construction of genomic DNA library. See here, first one. Uh, so see here, this weight and shown to the this diagram, and so you see that one. So first of all, what happened here? Construction of genomic DNA library. Here, double strand DNA structure is present. That one is after cleave the some enzymes, restriction nuclease enzymes are there. Restriction enzymes are there. So with that the help of restriction enzyme, these double strand DNA can cut into small fragments. Okay, Nandi. So cut into small fragments. So the fragments we will call as a genomic DNA fragment. So then these fragments are enter into a vector, into a particular vector. So what is that one? Insert the fragments into plasmids, mainly used the plasmids or bacteriophages. So that one recombinant plasmids are appear. So these introduce these recombinant plasmids into the a bacteria. So the bacteria then convert into the genomic to DNA libraries, means a number of fragments are produced in the host tissue. Okay. So next one is the construction of cDNA libraries. What is the complementary DNA library? So first one here also isolation of mRNA. When the construction of cDNA library starts with the isolation of mRNA from eukaryotic organisms. So first of all, we have to isolate the mRNA from the DNA, means messenger RNA, we have to isolate it. When we have to start the cDNA library, that one is a more first step, means this one is very essential to isolate the mRNA. So mRNA is isolated and purified using some different methods are there. Column purification method, mainly the column purification method we have to use that oligometric nucleotide coated resin that bind only the mRNA with polyacyl we have to use. The eukaryotic mRNA contain a polyacyl tail at the three point end. When the cell silate is passed through the poly T column, the polyacyl tail of mRNA molecule bind to the oligo DT sequence. This retains the mRNA in the column while all the other molecules that do not have polyetails pass through the column and are discarded. Finally, the mRNA molecules are separated from oligo TD sequence using an allocating buffer. So next one is the synthesis of cDNA. After isolation purification of mRNA molecule, and the next step synthesis of cDNA molecule from the isolated mRNA. At the first, a short oligo TD primer is uh, annealed to 3-polyethyl of mRNA molecule, which initiates the synthesis of first DNA using a reverse transcriptase enzyme. The primer is extended to form an RNA-DNA duplex. So the RNA is enzyme which is degraded the mRNA strands in the mRNA and DNA hybrid, leaving the small RNA fragments that are used as a primer for the synthesis of second DNA strand. The DNA polymerase 1 is the second DNA strand in segments removing the RNA primer. The DNA ligase enzymes seal the mix between the newly synthesized DNA fragments, resulting in the formation of a double-strand cDNA copy of the mRNA. Okay. So this is the synthesis of cDNA. Third step is cDNA cloning. The next step is the ligation of cDNA molecule into suitable vector for cloning. Since the cDNA has blunt ends and restriction site, linkers are adapted need to be added to the ends of cDNA molecule to make them compatible with the vector DNA. These linkers contain recognition sites for restriction enzymes. The linkers are then digested using restriction enzymes. The most commonly used vectors for cloning cDNA are plasmid and phase vectors. The suitable vectors are cut with the same restriction enzymes as the cDNA. The cDNA molecules are joined with the vector DNA, which creates the recombinant DNA molecule. So fourth step here, the transformation. What is here? The recombinant DNA molecule are transferred into host cells that can be culture to be produced colonies containing the cloned cDNA in cells. To select host cells that have successfully taken up the recombinant DNA, transformed cells are cultured on agar plate containing a selective medium. The selective medium contains antibiotics that inhibit the growth of untransformed cells. The presence of selectable marker gene on the vector ensures that only the cells containing the recombinant DNA survive and form colonies on the selective medium. The resulting colonies form the cDNA library. So another diagram is also there. This one is also see here. So see here. So see here, this one is the so human fibroblast means human cells are there. This first initially the human cells, fibroblast cells are there. So from that RNA extraction, and from that RNA extraction, here different markers are there. Total RNA, then cDNA synthesis, amplification, library preparation. So finally, we are to got the cDNA library, complementary library. See, another one is the screening a DNA library. After the construction of gene libraries, the screening process for both to cDNA and genomic libraries is important to identify and isolate the recombinant clones containing the DNA inserts of interest. So there are several screening methods are there. The most commonly used 344. So the most commonly used screening method for gene libraries is hybridization based screening method. This method used labeled DNA or RNA probes that are the complementary to target DNA sequence in the gene library. In hybridization based screening, the colonies are selected in the gene libraries are transferred onto a solid membrane such as the nucleocellular membrane. The membrane is then probed with the radio-labeled probe to identify clones containing the target sequence. The probe will hybridize with its complementary sequence in the target DNA. The positive clones are identified based on the presence of hybridization signals which are visualized using the autoradiography or fluorescence detection. Other commonly used screening methods include PCR screening, immunological assay, and sequencing based screening. So these are the screening of DNA library. So another applications of DNA library means genomic library and cDNA library. What is the application? So the application, what is the here? The genome libraries are essential for the constructing the physical maps of genomes, which helps in the understanding the layout and structure of genes. That is there. So this one is the helpful to understand the structure of genes. And these libraries are particularly useful for studying non-coding regions, regulatory elements, and gene sequences that may not be expressed. The genomic libraries from different species can be compared to study their evolutionary relationship. The genomic libraries also help identify genetic mutations and disease associated genes, which is the important for understanding the genetic basis of disease. This one is also most applicable. The disease uh, assessment of the identify of the genetic mutations are either genetic diseases. And what are the applications in the cDNA libraries? So what here the complementary cDNA means DNA libraries are useful for studying the actively expressed genes in different tissues under specific conditions. The cDNA library allow the identification and cloning of expressed genes. So this is very important application, that is the expressed gene different tissues under specific conditions. Another, the cDNA libraries can be used to compare gene expression profiles between the different species, which is useful in the study of evolutionary biology. So this is also another application. The cDNA libraries are also used to produce recombinant proteins. Recombinant protein structure also we have to study. These libraries are also used to identify and study the expression of disease relating the genes, which can help in the development of diagnostic markers. So these are the cDNA libraries, means genomic and cDNA libraries. Okay, clear? So, right. So coming to the uh, so next topic is the PCR, means the polymerase chain reaction. That is the technique is there, PCR technique. What is here PCR means? Wait, that's sometime. Wait. So, so what is the PCR here? So I'm shown to that technique one by one. We have to see. So, mm, okay, right. So what is PCR here? Polymerase chain reaction is a nucleic acid amplification technique used to amplify the DNA or RNA. In vitro enzymatically, it is a temperature depend on enzymatic process where either the specific targeted region of DNA or either whole DNA is replicated to quickly make millions of copies of the target DNA or DNA segment. It is called as a molecular photocopy. So it is a non-culture based nucleic acid, which is the cloning technique. We can quickly produce billions of copies of a single segment of DNA or RNA using the PCR technique. It is the temperature depending on the cyclic process, and by the end of the 20th to 25th cycle, over a million copies of the target nucleic acid segments will be produced in the technique. So it combines the principle of nucleic acid hybridization with the principle of nucleic acid replication. It denatures the double-strand DNA into a single-strand DNA, selected a segment of DNA to amplify using a specific primer, and then allow the method of replication using the DNA polymerase enzyme to amplify that segment. So PCR dates back to around in 1980s when the Carey Millis and his associate developed this revolutionary technique. He has awarded the Nobel Prize in Chemistry with Mitchell Smith in 1993. It has been most important tool in the molecular biology, genetics, and as the basis the tool of DNA and RNA analysis. So some objectives are there. What is objectives here? PCR technique to amplify the specific segments of DNA or RNA, resulting in billions of copies of a single DNA or RNA segment. To reduce the time required for the cloning of nucleic acid. Additionally, it has been modified to diagnose certain genes and mutations and support the other genetic and molecular studies or either techniques. Okay. The principle of PCR. What is the principle here? Polymer chain reaction. The PCR combines the principle of nucleic acid hybridization with the principle of nucleic acid replication with the temperature variation of the cyclic heating and cooling the so in these single-strand DNA templates, primers can anneal to their complementary sequence based on the nucleic acid hybridization principle. By continuously regulating the temperature, these three steps, that is the denaturation, annealing, and elongation, that can be permitted to occur in the cyclic manner, leading to millions of copies of single targeted nucleic acid sequences appear. What are the components of PCR? That is enzymes which are used in PCR. That is the nucleic acid template, means template DNA. So template DNA is a sample DNA that containing the selected nucleic acid sequence that needs to amplify. The template must be DNA only. The genomic DNA, complementary DNA, and plasmid DNA. The reverse transcriptase polymerase chain reaction used RNAs as starting material, but RNAs are primarily converted into complementary DNA before amplification. The template DNA must be highly pure with an absorption ratio of that is 1.8. A quantity of zero point 0.1 to 200 millimicrons can be used with the ideal quality of 30 to 50 microgram. So see here that the diagram is there, the DNA molecule is there, the DNA molecule, then DNA polymerase enzyme, with the DNA primer, and we add to PCR buffer. That PCR machine is there, and PCR machine here. So next to the DNA polymerase are enzymes that is the catalyze the synthesis of complementary DNA starts by assembling the nucleotide sequentially according to the template stance. Simply, it is the enzyme that synthesis DNA, hence the plays key role in the DNA replication. That tag the DNA polymerase enzyme extracted from the bacterium, that is the its most widely and best known DNA polymerase used in the PCR since it is established. TAQ means TAQ DNA polymerase is the thermally stable and continue its actively after the repeated heating and cooling cycle. It is stable up to 95 degrees and shows the most effective reaction at around 72 to 78, incorporating about 60 base per second. In 50-liter reaction mixture, around 1 to 2 units of stack polymerase is sufficient for the amplification. So the most DNA polymerase enzymes are available. That is the Vent enzyme isolated by the Thermococcus litoris, and PFU enzyme which is isolated from the Pyrococcus furiosusum. Another one is the primers. Primers are officially synthesized short single strand sequence of oligonucleotides that are the complementary to the targeted nucleic acid sequence in the template. The DNA short sequence of around 15 to 30 bases that act as a starting point of DNA synthesis. They annually at their complementary position in the single strand template DNA strands. The DNA polymerase enzymes then extend this primer from its OH end, forming a new complementary strand. Usually 10 to 12 pmol of each primer is sufficient for a PCR reaction. The PCR primer are of two types, forwarded and reverse primer. The forwarded primers are complementary to the antisense strands and are responsible for the amplification of the antisense strands. They are called 5' primer. So the reverse primer are complementary to the sense stand and are responsible for the amplification of sense stand. They are also called as a 3' primer. Nucleotides, fourth one is the nucleotide. Deoxynucleotide triphosphate are artificially synthesized nucleotides that act as building blocks of a new DNA strand. There are four different deoxynucleotide triphosphates used in the PCR. That is the deoxydenosine triphosphate, deoxyguanosine triphosphate, deoxythymidine triphosphate, and deoxycysteine triphosphate. These four deoxynucleotide triphosphates are sequentially added to the unlead primer by the DNA polymerase enzyme, generating a new strand of DNA complementary to the template strand. The PCR buffers and other chemicals we have to use. All the process need out their trice HCl based buffer system of pH around 8 to 9.5. The common buffer used anodic buffer with the additional magnesium chloride. Common components of the PCR buffers are dimethyl sulfide and ammonium sulfate, non-conic detergent, and polyethylene glycol. These are the some tetramethyl ammonium chloride, EDTA, ethylene diamine tetraestic acid. So these are the buffer system increase the reactions efficiency and specificity and prevent the inhibition and secondary structure of formation during the process. So another one is the thermocycler that is known as the PCR machine. So thermocycler is simply an electrical heating device that regulates the temperature as per need during each stage of the PCR process. This machine increase the temperature during the denaturation steps and lower it during the annealing and again increase it during the elongation step. This process of increasing and decreasing the temperature occur in a cyclic manner according to the pre-programmed setup or instruction by the user primer to open. So some steps which are used in the PCR. So there are three core steps in any type of PCR: denaturation, annealing, and elongation. These three main steps are there. Apart from these three primary steps, the preparation phase at the beginning and PCR product analysis phase at the end are also important steps. The whole step can be summarized into three steps, that is the pre-preparation, application, and product analysis. Pre-preparation, it is the initial step before the actual polymerase chain reaction takes place inside the thermocycler. In this step, one must prepare a reaction mixture and load it on the programmed thermocycle in order to amplify the target DNA or RNA segment. The sample DNAs or RNAs are extracted from the sample and stored. All the material are arranged safely. Measure are taken. The PCR reaction preparation area is cleaned. All the reagents are brought into working temperature. The sample is extracted or brought from the storage. The PCR reaction mixture is prepared. The thermocycler is programmed and reaction mixture is loaded on the thermocycler. So I shown to another diagram. So this one. So here the steps one by one we are to see. And the next one is the amplification. This is the main reaction process occurring in the PCR. The amplification step includes denaturation, annealing, and elongation occurring orderly in the cyclic manner, one after another for the certain number of cycles pre-programmed by the user. Step one is the denaturation and annealing and elongation. So it is the first step. What is that one? Denaturation, amplification reaction, where the double strand DNA is thermally denaturated into two single-stranded DNA templates. The temperature is raised about to 94 degrees for 30 to 90 seconds. At this temperature, the thermal energy overcome the weak hydrogen bonds holding into two DNA strands together, allowing them to separate single strand DNA to double strand DNA to single strand templates. And another one is the second step is annealing. So denaturation is followed by the annealing step where the primer anneals are single strand DNA templates at their complementary sites. The forwarded primer anilines at the complementary site of the ancestance stands and the reverse primer anilines at the complementary site of the sense stands of the template DNA. For annealing to occur, the temperature is reduced to 57 to 70 annealing temperature and the differ based on the G content of the primer. So another one is the elongation. It is the final step in the amplification reaction where the temperature is raised to 72 degrees so that the tag DNA polymerase enzyme is begins synthesizing the new DNA strands in the 5 to 5 to 3 direction. Array. The DNA polymerase enzymes added nucleotides from the reaction mixture to OH end of the annealed primer forming a new complementary strand. The time is required for elongation depends on the same nucleic acid sequence length and DNA polymerase activity. Generally, elongation takes place at the rate of 1 kilobase pairs per minute. So product analysis phase, it is the phase after completion of the PCR where the reaction mixture subject to PCR is analyzed to confirm that desired amplification is achieved. The mostly agarose gel electrophoresis is employed in order to check for amplified DNAs or RNAs. However, no additional step is required in some types of PCR like real-time PCR. So some different types of PCRs are there. Assembly PCR, LU-PCR, and conventional PCR, cold PCR, colony PCR, digital PCR, droplet. Some different types of the PCRs are there. So what is the application of PCR? So identification and classification of organisms, some different molecular biology, some different applications are there. So PCR is used widely in identifying the microorganisms up to level of subspecies and chains level also. So DNA isolated from fossilized animals also amplified and studied to relate them with the animals that are still living on the earth And another one is the infectious disease diagnosis. The use of PCR is the identification of pathogen has lead to the quick and accurate diagnosis of infections. Not only diagnosis, but parallel identification of antimicrobial resistance gene in the pathogen is also possible. Another application, some gene mutations and genetic disorders we have to identify with the PCR. So mutation in any segment of the DNA can be detected by using PCR. So knowing this mutation, we can confirm a genetic disorder. DNA polymorphism can also be analyzed, which can also related to a genetic disorder of some kind. In the PCR is used one of the most important tool to diagnose conventional diseases, genetic disorders, and any other mutation leading to negative health problem. So detection of cancerous cells is another very important application in the PCR. DNA fingerprinting also. So PCR is used for DNA fingerprinting. DNA fingerprinting is used for the identification of criminals and individuals and for confirming the parents. Gene sequencing, identify the gene sequencing also. The large number of using techniques like PCR, all the sequencing method of PCR are their important step. Another one is the DNA and RNA quantification. PCR can also be used for the quantification of sample DNA or RNA. Another one is that this PCR is very important tool in the genetic engineering. So this one is very used in the genetic engineering for analyzing the modified DNAs and amplifying the target or vector DNA. So this is the some. What are the advantages? Some advantages. What are the advantages are there here? It is a very fast method for producing the multiple copies of nucleic acid segments within a few hours, few days. Billions of copies of specific nucleic acid sequence can be produced and analyzed by the PCR. So this one is the most important. It can be analyzed very minute quantity of DNA or either RNA. It's the principle and produces are the simple to understand and applicable. Being a semi-automatic technique, the process is operated and regulated automatically by the thermocycler. It has shortened the time for the diagnosis of diseases and genetic disorders. It is the base for several molecular techniques like DNA fingerprinting, genetic imprinting, and genetic engineering, gene sequencing, and molecular vector production. So microarray is also another application advantages in the PCA. So some limitations also there, disadvantages also of the PCR. The prior information regarding the sequence of DNA or RNA target segment is required. Even very small contamination in the sample can be amplified, it giving the false or negative result. This is a chance of mutation during the process of DNA polymerase enzyme makes any error during the replication. So PCR fails to detect the novel mutation, multigenetic disorder, and structural and numerical chromosome abnormalities. So most type of PCR process require additional steps for the product analysis. Is the complex is requiring the sophisticated technology, a wide range of chemical and highly trained professional to operate this one. So it is not suitable for very long DNA molecule. Very long DNA need to be cut into smaller fragments. So RNA needs to be first converted to DNA using reverse transcriptase enzyme before its amplification. So these are the some PCR technique, PCR introduction, and what are their remaining? Mm, mm,